Okay, so in terms of bacteria, I do want to look at some of the importances to humans. So we'll look first at the medical importance of bacteria. So at the beginning, I mentioned to you that bacteria can cause disease. Those are the bad guys, okay? And so we call these bacterial cells pathogens, okay? And really, pathogens can cause problems in several ways. One is that they might be producing toxins. And so if they're living on you or in you and they're producing toxins, that's going to cause disease, okay? They can also attach to surfaces or organs and cause problems within those organs or on the surface of your skin. And if they're doing that, then that can cause disease too. So those are a couple of ways where we would say, well, these would be bacteria that are kind of like the bad guys and they're causing problems. Okay, so in terms of pathogens, sometimes we need to use an antibiotic to kill off those bacteria. And so the main way that a lot of the antibiotics work is by rupturing the cell wall. So if you can break apart the cell wall, you're going to kill that bacterial cell. So I put a video here for you to watch. I'm not going to play it right now, but when you're looking through the PowerPoint, I'd like you to watch, and it just kind of shows bacteria that have been exposed to antibiotics, and that you just start seeing these cells exploding because their cell walls are being ruptured. Okay, but there are also antibiotics that are chemicals that would interfere with bacterial metabolism, which would be all of the reactions that are happening within that bacterial cell. And normally their metabolism is different than ours. They use different enzymes than we use. And so if you can interfere with an enzyme that's important to a bacterial cell, you're gonna cause that cell to stop functioning correctly. Just very briefly here, in terms of medical importance, there are bacteria that can cause sexually transmitted infections, okay? Some people show some really gross, horrible pictures of sexually transmitted infections. I'm not going to do that, but just a couple of things. I would like you to know what kind of infections are caused by bacteria. And if you know that, then you would know that then they would be also treatable because you can take an antibiotic. So chlamydia is a very common bacterial sexually transmitted infection. And if you are diagnosed with that, you will be given an antibiotic that will clear it up. Pelvic inflammatory disease is a little bit more general because I've got a picture here of the female reproductive system, but it could be something that's infecting or causing inflammation in the vagina or the cervix or the uterus, or maybe in the fallopian tubes, or maybe in the ovaries. And so bacteria can pretty easily get into the reproductive system through the vagina, and if they end up taking up residence in there and they overgrow, you're going to have infection. And so pelvic inflammatory disease is just kind of a general term to refer to any of those areas that might be infected. The other two that you've probably heard of, and again, I'm not showing gross pictures here, but gonorrhea and syphilis are both bacterial diseases, meaning that if you catch it early, you can treat it with an antibiotic and it's going to be okay. I'm just looking at graphs here, and they're not super current. They go up through 2005, but you see that there, was kind of a high spike in the 70s, both for syphilis and gonorrhea, but they've come down quite a bit in recent years. Just so you know, gonorrhea causes a lot of inflammation and a lot of extra fluid production. And so one of our professors used to work in public health, and she said, when people would come in with gonorrhea infections, especially men, it was called the baggy disease because basically so much extra fluid produced because of the infection that they had to come in with a baggie over their penis because of all of the extra fluid. So, but if that's caught early, you treat it with an antibiotic and you're going to be fine. If you don't catch it early, you don't treat it, there are some medical problems that occur as a result of that. Okay, so we have talked a little bit about antibiotic resistance, and I think I probably mentioned the term superbug, but basically that's a common name term that's referred to bacteria that are highly antibiotic resistant. Okay, and so that means that it's very difficult to treat them because they've picked up genes that make them be able to withstand antibiotics. And so some of the reasons for this is that in our country we have really overused antibiotics. People have begged their doctor to give them an antibiotic when they have some kind of a viral infection that really can't be treated with an antibiotic, and that ends up wiping out good bacteria and only leaving some of the bacteria that are pretty pathogenic, and they become more pathogenic as they pick up more new genes. Also, antibiotics have been used in agriculture, you know, with the chickens and the pigs and things like that. They're treated with high doses of antibiotics to try to keep them healthy, but those things end up creating populations of antibiotic resistance. And then, of course, mutations can cause antibiotic resistance too. So there's two videos that I've put at the bottom here that I really would like you to watch. The killer microbes is one on how some of these really highly resistant strains have evolved. And then the second one is a really interesting study from Harvard. It's just about a 3-minute video, but it shows how bacteria can evolve and pick up mutations in the course of, 10 days. And so both of them are kind of interesting in regard to antibiotic resistance. So I do want you to take a look at those when you download your PowerPoint. Okay, so I mentioned that that bacteria are very biologically successful, meaning that they can really live almost anywhere and adapt to the environment. And part of the reason that that's the case is because when environmental conditions are bad, whether it's super high heat or dry or extreme cold or any of these things that might make it hard for bacteria to survive, what they do is they form endospores, which basically is like they go into hibernation. And then when the environmental conditions get better, then they come out of the endospore and they're still viable. So in the top picture you can see the endospore on the outside of the cell. In that middle picture with the purple, the gram-positive bacillus, you can see the clear little circles inside the cell. That's the endospore. Okay, and so those endospores can remain dormant for a very long time, and when they come out of it, they're still virulent, which means they can still cause disease. So they've found, you guys have heard of anthrax probably, they've found anthrax spores that are over 1,000 years old that when they put them in the right conditions, they are able to grow again and cause disease. Same thing with the bacteria that causes botulism. So those are just some examples of why bacteria are so biologically successful. Just a point on botulism. Botulism is caused by Clostridium botulinum, which is a bacterial species. And when you get botulism and you get these terrible stomach cramps and so forth, it's because of a neurotoxin that's being produced by these bacteria. And that neurotoxin is the basis for Botox, so botulinum toxin. And so basically Botox is this very highly effective neurotoxin which causes your muscle cells to basically be paralyzed, and that gets rid of wrinkles. So what I'm saying is that Botox, although people have doctors have perfected the use and all of that kind of thing. But to me, that's a little bit of a scary situation. Okay, so environmental importance. So we've talked about the fact that bacteria can be decomposers, and there are a lot of them. And so we mentioned that they would be saprotrophic decomposers. They're decomposing or digesting breakdown and waste products outside of their body and then absorbing what they need, and everything else is recycled, meaning it's put back into the environment to be used by plants and whatever else is in the environment. Okay, so in the pictures at the bottom, they're pretty high magnification pictures, but you can see roots and soil, and all of them have bacteria around them. Okay, so I put in 2 short decomposition videos, which I think are good for kind of reinforcing this point right here, so make sure that you watch those. And then bacteria are used in sewage treatment plants, and so they're used in processing and breaking down human waste as sewage. So I don't know if you've ever, there's a sewage plant in Arlington, and if you drive by that sewage plant, it doesn't smell that great, but what's happening is bacteria are taking waste and breaking it down, and of course as they do that, they're producing gases that have kind of noxious smells. Okay, and they can also be used to degrade pollutants, so things that end up in the environment that are pollution in the environment. There are bacteria that are adapted to eat oil, and so if you can spray bacteria, oil-eating bacteria on oil spills, you're not putting detergents in, you're not putting chemicals in the environment, you're putting in bacteria that actually can do that job for you, which is much better for the environment. So your book has a couple of pictures of bioremediation, which would be remedying the biological environment using a biological organism. Okay, and then we have products that are made using bacteria. So I think you know that alcoholic beverages are made by fermentation, and many times the fermenter is some type of bacteria. Cheese is made by fermentation. We have bacteria that have received genes that help them produce vitamins. We have bacteria that grow and produce their own antibiotics for other bacteria. Yogurt is fermented. And so bacteria are very useful and produce products that we can use. When we talk about biotechnology, basically if we're going to engineer bacteria, we're going to be putting in genes that help them to produce different chemicals or drugs that might be useful as a pharmaceutical or something like that. So bacteria have an important economic role also. Okay, so your book has just pictures of the sewage treatment, of manufacture of cheese, and then production of insulin now, which is a hormone used for type 2 or type 1 diabetes. It's way cheaper to have a bacteria pump it out for you. It's a protein, so you can give bacteria the gene for making insulin, and you can produce human insulin very cheaply that way. Okay, so I kind of like this kind of overall big picture diagram from your Hofnagel's text on where you find these different kinds of bacteria and what some of their functions are. So just in this one picture of somebody sitting eating yogurt next to a lake or a pond, you have skin bacteria, you have digestive tract bacteria, you have bacteria in the yogurt, you have decomposer bacteria, cyanobacteria, which are autotrophic, and you have some nitrogen fixing bacteria. So there's a whole lot of them there, and in that case all of those are beneficial bacteria. They're all the good guys. Okay, so the last thing we have in this chapter is to look at the domain archaea, which is also a prokaryotic domain, but it is more closely related to the eukaryotes than the bacteria. So we show the domain archaea on the same branch as we see the domain eukarya, which is where we would be organized. Okay, so they have some differences in the way their cells function in that they're able to survive pretty extreme conditions. There's bacteria that can too, but the archaeans can survive in hot springs over 100 degrees and around volcanoes. They can survive in places with no oxygen, places like that where you wouldn't necessarily expect living organisms to live. You're going to be able to find archaeans there. So sometimes we call them extremophiles, extreme referring to the conditions, and philos meaning love. And so they're extreme lovers, okay? Part of that is because they have an interesting cell membrane that's able to function in very high temperatures. So we do find some of them as chemoautotrophs, meaning they can make their own food using chemicals. So we don't find them as photoautotrophs. They don't have chlorophyll, but we do find those that are able to make their own food. Okay? And usually we classify them by habitat. Where would you find them living? And so I'm just going to look at a couple of examples of habitat. Okay? We have those that are classified as methanogens, meaning that they're producing or generating methane, but many of these you find in anaerobic environments. So swampy areas down in the mud, those places would be anaerobic, and that's a place where you would find these methanogenic archaeans. We have those that are halophiles. So halo refers to salt and philos refers to love. So these are the salt lovers. And it's not just that they like salt, but they actually require high concentrations of salt. So we're talking about salt pits and stuff like that, or maybe the Dead Sea. The ocean, that concentration of salt is not considered a super high salt concentration environment. Okay? And then I put these two together, but we could separate them. So an organism that's a thermoacidophile means that it likes hot, very hot environments and acidic environments. But we also have those that would just be thermophiles or acidophiles, and sometimes you find them together. So this is where we're talking about the hot springs and thermal vents and volcanoes. This is where you would find these archaeans living. Okay, so kind of as a big picture wrap up, we looked at a bunch of different prokaryotic cells. We looked at the two domains. We talked about how they reproduce by binary fission. We looked at acquiring new genes, and in this case this would be horizontal gene transfer if you're talking about conjugation, transduction, and transformation. If you're talking about binary fission, that would be vertical gene transfer. And then we looked at some of the characteristics that would be used to classify your different types of bacteria. Okay, so in terms of what we've looked at, we've looked a little bit at evolution and speciation, and then we're starting to look at living organisms, starting with the two prokaryotic domains. So the next group of organisms that we'll look at is the protist kingdom, which is in the eukaryotic domain.